Welcome to today's Explainer Focus Learners. Today, we're injecting some serious energy into unlocking one of the most fascinating secrets of human biology, how medicine actually works once it's inside our bodies. We're going on a journey of microscopic border control, and trust me, it's full of tiny barriers, bouncers, and VIP molecular pathways. Think about it for just a second. How does a tiny pill, once you swallow it, know exactly where to go to cure your headache or lower your blood pressure? Well, the truth is, the human body is an incredibly secure fortress. It absolutely doesn't just let any random chemical wander around freely to its destination. So to understand how medicine pulls off this microscopic heist, here is our addenda. 1. The cellular border wall. 2. Anatomy of the membrane. 3. Crossing the divide. And 4. Transporters and toll booths.
Let's jump into Section 1, the cellular border wall. Okay, let's dive into this. To effectively treat the body, every single drug has to first figure out how to bypass our natural microscopic border controls. From the very moment a drug enters, it's interacting with cell membranes continuously. This happens during absorption across the gut into your circulatory system, and it happens again when passing through capillary membranes to distribute to targeted tissues. It's literally an endless cycle of crossing borders. We capture this entire journey using a really critical acronym in pharmacology. ADME. That stands for absorption, distribution, metabolism, and elimination. This cycle defines a drug's complete lifespan inside your biological membranes. It's the fundamental blueprint for drug design, dictating exactly how long and how well a medicine will actually work in your system. Moving on to Section 2, anatomy of the membrane. Let's zoom all the way in from the macro tissue level. Drugs don't just cross one simple flat wall. They often have to penetrate multiple layers of epithelial cells lining our organs and body cavities. To get from the lumen, say the apical side inside of your stomach, to the basolateral side and into your blood vessels, they have to navigate a highly complex 3D cellular landscape packed with tight junctions and transmembrane proteins. Now, what's really interesting about this barrier is its molecular structure. The membrane perfectly guards the cell using a lipid bilayer made of phospholipid molecules. Each of these molecules has a hydrophilic, or water-loving polar head facing the outside, and a hydrophobic, water-fearing, nonpolar fatty acid tail hiding on the inside. It creates this brilliant chemical sandwich that naturally repels most foreign substances trying to enter. And hey, this wall isn't completely static. While the packing of fatty acids can be quite fluid due to a mixture of saturated and unsaturated tails, cholesterol acts as a critical, rigid stabilizer. Because it's amphipathic, it packs really closely with the phospholipids to create a liquid, ordered state. This keeps the membrane from becoming a rigid gel, ensuring stability and directly impacting how well a drug can penetrate those lipid rafts. Section 3, crossing the divide. So now that we know this wall is made of tricky phospholipids, how does a chemical actually sneak across? Well, it depends heavily on its chemical properties, specifically whether it's hydrophilic, hydrophobic, or amphiphilic. Gases and small hydrophobic molecules like benzene, they zip right through that lipid core, but large polar molecules like glucose or fully charged amino acids and ions just bounce right off. They simply don't have the right chemical disguise to slip past the hydrophobic tails hiding in the center of the membrane. For the drugs that actually can slip through, they usually do so via passive diffusion. And of course, there's a mathematical equation that dictates the exact rate of this movement. But hey, don't let the calculus intimidate you. This equation really just calculates the speed at which a chemical moves down its concentration gradient across the membrane. And that happens to be the most common form of drug movement in the body. So the crucial point here is that this intimidating math really just boils down to 3 super intuitive factors. First, the concentration of the drug, how much of it is waiting at the border. Second, the drug's permeability, which reflects how fast that specific chemical can cross. And third, the surface area of the membrane it's trying to cross. More drug, higher permeability, and more cellular space to cross equals a much, much faster cellular infiltration. Even within passive diffusion, drugs have options. They can take the transcellular route, literally plunging straight through the cell itself, which relies heavily on the drug's lipophilicity. Or they can take the paracellular route. This is where small, partially charged drugs try to slip through the water-filled junctions between the cells. The success of paracellular transport totally depends on the tissue. Like, your gut and skin junctions are very tight, but the epithelium junctions in your nose are much looser, making for a way easier crossing. Which brings us to Section 4, transporters and toll booths.
So, what happens when a drug just can't passively diffuse because it's too large or too charged? It has to use the guarded gates of the cell. Lipid-insoluble solutes require a specific protein partner, acting kind of like a cellular usher. This usher physically binds to the chemical and creates a channel to facilitate its movement down a concentration gradient, right into the cytoplasm.
But it's not just a friendly one-way street. There is a constant tug of war happening at the border. On one side, we have influx, or uptake transporters, actively inviting drugs into the cell or bloodstream. On the flip side, we have efflux transporters. Think of these guys as the cellular bouncers. Proteins like the ATP binding cassette family are specifically designed to grab foreign chemicals and forcefully kick them back out of the cell to protect the body.
Sometimes a drug needs to be pushed against the crowd, what we call moving against the electrochemical gradient. This requires active transport. While passive transport is an easy downhill slide from high to low concentration, active uphill transport requires actual energy. These active transporters use primary energy like ATP or secondary energy to pump molecules against the gradient.
Let's look at some of these specific VIP lanes, like the solute carrier superfamily. These secondary active transporters come in a few different styles: uniporters, symporters, and antiporters. For example, the OCT and OAT families are specialized proteins that explicitly transport organic cations and anions. OATs act as antiporters, meaning as they bring one anion in, they send a different coupled ion out, creating a perfectly balanced molecular exchange across the membrane. Let's see how all this builds into a precise mechanical sequence. Step 1. A substrate-specific drug docks at a given transporter. Step 2. The protein literally undergoes a physical, structural change to move the drug. Step 3. The drug is released to the intracellular space. But here's the major catch. These transporters are finite in number. If too many drug molecules are present, these gates become totally saturated, meaning drugs literally have to compete against each other for access. And this microscopic border battle is not just happening in one isolated place. It is happening simultaneously across your entire body, across your intestinal epithelium for absorption, inside your liver's hepatocytes for metabolism, and across your renal tubules for excretion. It's a massive, coordinated network of influx and efflux transporters like PBT, OCT, and PGP that ultimately dictates exactly how medicine acts in your body.
Which brings us to our final provocative thought. The cellular membrane is an absolute masterpiece of biological engineering. But as pharmacology advances, we're getting better and better at designing molecules that bypass, hack, or perfectly partner with these barriers. If we can truly master the membrane, what biological barriers can we break next? Could we seamlessly deliver cures directly to the brain, or target individual diseased cells while leaving the rest of the body completely untouched? Thank you for joining me on today's Explainer Focused Learners. Keep studying, stay curious, and I'll catch you in the next one.